Good morning, my dear students. We will be discussing on mitochondria, chloroplast, ribosomes, and nucleus. First, let us take up mitochondria. They are glandular or filamentous organelles present in the cytoplasm of animal and plant cells. They are known as powerhouses of the cells as they are the main site for the generation of high-energy phosphate bonds ATP through oxidative phosphorylation. Allmutt first observed them as bioflats at the end of 19th century, but Benda in 1897 called these structures as mitochondria. The shape, size, and number of mitochondria vary in different cells. The shape of mitochondrion in general is filamentous or granular, but during certain functional stages, other derived forms may be observed. The size of mitochondria varies with diameter ranging from 0.2 to 1 micrometer and length from 2 to 10 micrometer. Mitochondria are concentrated near the spindle during mitosis, but they are distributed appropriately in equal proportion between the daughter cells after the cell division. A normal cell may contain 200 to 300 mitochondria, but its number may vary considerably from a few to 1,000 or more depending on the cell type and condition. Mitochondrion consists of an outer and an inner membrane with a thickness of 60 angstrom. The outer and inner membranes are separated from each other by a space of 80 to 100 angstrom. The inner membrane forms many finger-like projections or infoldings which are called mitochondrial crest or cristae. This inner membrane divides the mitochondria into two chambers or compartments. The outer membrane existing between the two membranes and in the core of the crest, and the inner membrane which is filled with a relatively dense proteinaceous material usually called the mitochondrial matrix. The matrix is usually homogeneous, but in some cases it may contain a finely filamentous material or small, highly dense granules containing phospholipids. Small ribosomes and naked circular DNA are present in the matrix. The mitochondrial matrix also contains soluble enzymes of the Krebs cycles, RNA, and other components for protein synthesis in the mitochondrion. The exterior of the outer membrane is made up of small stalkless spheres packed closely together to give a rough appearance of the mitochondrial surface. The inner membrane, on the other hand, has tennis racket-like particles of 70 to 100 angstrom on its surface. These structures are called elementary particles or F1 particles. Each particle has a stalk of about 50 angstrom long and 30 angstrom wide and a head of about 30 angstrom in diameter. The entire length of the structure is about 160 angstrom. The F1 particles are regularly spaced at interval of 100 angstrom on the surface of the inner membrane. The F1 particles are previously thought to contain all the enzymes of electron transport chain and are therefore called electron transport particles. Recker in 1967 had shown that these particles contain ATPase and were involved in the process of oxidative phosphorylation during respiration. There are about 10 to the power 4 to 10 to the power 5 elementary particles present in a mitochondrion, and the presence of this particle on the matrix site confers to the inner membrane a characteristic asymmetry that is of fundamental importance to its function. The mitochondria have been shown to possess the ability to catalyze the reactions of the Krebs cycle using pyruvic acid as fuel. The pyruvic acid is completely oxidized to carbon dioxide and water with the release of energy. Now let us come to chloroplasts. Plastids are macroscopic organelles found in eukaryotic plant cells. The most important plastid in plant is the chloroplast, which are characterized by the presence of important green pigment called chlorophyll. They are involved in the process of photosynthesis. The shape, size, and number of chloroplasts may vary in different cells within the species. The shape of chloroplast may be spheroid, ovoid, or discoid, but may be frequently vesicular with colorless centers. The chloroplasts have an average diameter of 4 to 6 micrometer in higher plants, but the size may vary according to sexual and genetic differences. The number of chloroplasts in higher plants are 20 to 40 per cell, but in algae like Chlamydomonas, there is only one huge chloroplast. The chloroplast has three main components: the envelope, the stroma, and the thylakoids. The chloroplasts are enclosed by a double-layered semi-permeable membrane called envelope of about 150 angstrom thick. The stroma fills the chamber enclosed by envelope and is a kind of gel fluid phase that surrounds the thylakoids. It contains chloroplast proteins, ribosomes, and DNA. 50% of chloroplast proteins are present in the stroma. Carbon dioxide fixation and synthesis of sugar, starch, fatty acids, and some proteins take place in stroma. The thylakoids are either stacked like a pile of coins forming thylakoid grana, or may be unstacked producing stroma thylakoids. The stroma thylakoids forms a system of anastomosing tubules connecting different grana thylakoids. The thylakoids are composed of proteins and lipids, and the chlorophyll pigment is inserted in between proteins and lipid portions. The size and number of grana in chloroplast varies in different species, but generally there are 20 to 100 grana in each chloroplast. Each granum is about 0.4 to 0.8 micrometer in diameter and about 0.3 to 1.7 micrometer in thickness. The stroma thylakoid is much thinner as compared to grana thylakoid and contains unpigmented lipoprotein and associated enzymes which are involved in secondary reactions of photosynthesis. Chlorophyll, carotenoid molecules, and a reaction center are assembled in thylakoids forming two photosystems, 1 and 2. Each photosystem is associated with an electron transport system and with structural proteins. Lipids present 50% of thylakoid membrane, and these include those involved in photosynthesis. The membranes of thylakoid are composed of many paracrystalline particles called quantosomes, which are photosynthetic units and are aggregated together to form a larger particles of 500 into 100 angstrom. The thylakoid membrane also contains 9 nanometer spheres which are protruding from the outer surface and are considered as enzyme complex ATPs. They are known as coupling factor, very much like the F1 particles found on the inner membrane of the mitochondria. The chloroplast is a site of photosynthesis which requires specialized spatial arrangement of chlorophyll, associated pigments, and of enzyme systems for effectively trapping light energy and storing them into compounds from which other forms of energy may later be derived for the benefit of the organism concerned. Now let us take up ribosomes. Palette first observed ribosomes under electron microscope as dense particles or granules in the cytoplasm of cell. This organelle is present in prokaryotic and eukaryotic cells and is the site of protein synthesis. There is considerable variation in the number of ribosomes present in prokaryotic and eukaryotic cells. There are about 10,000 ribosomes in a single prokaryotic cell, while the number increases considerably in eukaryotic mammalian cells, which may contain about 10 million ribosomes. Both the ribosomes types, prokaryotic and eukaryotic, share the same general structure and organization. The ribosomes of both the cell types are made up of RNA and proteins. Ribosomes are basically ribonucleoprotein particles. The intact prokaryotic ribosomes is termed as 70S ribosome, which is made by association of two unequal subunits, the smaller 30S and larger 50S subunits. The proteins and the RNA are present in ribosome in approximately equal amounts. The 30S subunit is made up of 116S RNA and other 21 different proteins. The proteins of the 30S subunits are termed as S1, S2, S3 and so on. S denotes for small subunit. The larger 50S subunit is composed of two RNA molecules, that is 5S ribosomal RNA and 23S ribosomal RNA, along with 31 different proteins. The proteins of the 50S subunit are denoted by capital letter L for the large subunit. Most of the proteins up to 34% are very basic proteins, for example arginine and lysine, which may be the reason for their strong association with RNA, which is acidic. However, the positive charges generated by the basic amino acids are not sufficient enough to balance the many negative charges in the phosphates of the RNA. This is a reason for strong negative character of ribosomes and they bind cations and other basic dyes. The ribosomal RNA has high degree of secondary structure, forming hairpin loops between the complementary regions of the same linear RNA molecule. The ribosomal structure is maintained when the ribosomal proteins adhere at the specific points on these RNA loops. The basic features of eukaryotic ribosomes are similar to those of prokaryotic ribosomes, though they exhibit more complexity in terms of larger protein size and higher number in addition to one extra RNA molecule. A typical intact eukaryotic ribosome is ITS, which comprises of two subunits, a smaller 40S and larger 60S subunits. These sizes may vary over a 20% range from one organism to the next, in contrast with bacterial ribosomes, whose sizes are nearly the same for all the bacterial species examined. The 40S subunit is composed of one 18S ribosomal RNA and about 30 proteins, while the larger 60S subunit is made up of one 5S ribosomal RNA, one 5.8S ribosomal RNA, and one 28S ribosomal RNA along with about 50 other proteins. Now we have nucleus. This is the most important component of a cell, which is enclosed by specialized double membrane structure. It controls the cellular metabolism and contains all the genetic materials. Leeuwenhoek first observed nucleus in the red blood corpuscles of fish, but Robert Brown was the first to study nucleus in orchid root cells in 1831. Most of the cells of plants and animals are uninucleate, that is, they contain single nucleus. Some cells, however, contain two nuclei. For example, cells which are present in protozoan like paramecium. The nucleus is usually placed at the center of the cell, though the position may vary according to the cell type. The nucleus is located at the peripheral position of the plant cell due to the presence of large central vacuole. The shape of nucleus is generally spherical in shape, but it may be of ellipsoid or flattened in some other cells. Although the shape of nucleus is constant, the size may vary at different times during the cycle of cellular activity. The chromatin material present in the nucleus does not change and remain constant for a particular species, even though the shape and size of nucleus may change in a cell. The nuclear envelope encloses the nucleus, demarcating it from the cytoplasm of the cell. It is composed of two membranes. Each membrane is about 75 to 90 angstrom thick and lipoprotein in nature, and they are separated by space of 100 to 150 angstrom called the perinuclear space. The outer nuclear membrane may be smooth or rough when ribosomes are bound onto it, but the inner membrane is always smooth and contain no ribosomes, but sometimes associated with chromatin. The nuclear membrane is broken at regular intervals by nuclear pores, which are octagonal orifices about 80 nanometer in diameter. The nuclear envelope regulates the passage of ions and macromolecules. The nuclear pores present in the envelope play an important role in the transfer of macromolecules between the nucleus and the cytoplasm and vice versa. Thread-like, coil, and much elongated structures are present inside the nucleus of cells at interphase stage. These thread-like structures are called chromatin or nuclear reticulum, and they are a complex of DNA and histone proteins. But the chromatin fibers become thick, short, and ribbon-like structures called chromosomes during cell division. The chromatic fibers are distributed throughout the nucleoplasm and are differentiated into two regions. Euchromatin is narrow, about 10 to 30 nanometer thick, lightly stained and diffuse part which form the bulk of chromatin. It, however, takes deep stain during cell division and contains maximum DNA and is genetically very important. Heterochromatin is wider, about 100 nanometer thick and coiled tightly. There is a greater density of chromatin material and thus tend darker in interphase nucleus. This region is genetically inert as it contains relatively little DNA and large amount of RNA. Nucleoplasm or chiroclamp is a transparent, semi-fluid and colloidal substance which fills the space between nuclear envelope and nucleolus. It is also termed as matrix, in which the chromatin thread and nucleolus remain suspended. Nucleoplasm is mainly composed of nucleoprotein and it contains various inorganic and organic substances such as nucleic acids, proteins, enzymes, etc. The nucleolus is large spherical body found inside the nucleus and first discovered by Wagner in 1882. The number of nucleolus present in a nucleus varies with species. Nucleus of some cells of plants and animals may contain one or two nucleolus or even more. In human, there is two pair of nucleoli in its diploid nucleus. The nucleolus is chemically composed of proteins and nucleic acid, that is DNA and RNA. The RNA present in nucleolus is closely similar with the ribosomal RNA. It is believed that nucleolus helps in the synthesis of ribosomal RNA. The DNA present in the nucleolus guides the synthesis of RNA. The RNA after synthesis passes from the nucleolus to the nucleus and then to ribosome in the cytoplasm. Now let us come to the conclusion. Mitochondria, chloroplast, ribosomes, and nucleus are the main organelles of eukaryotic cells having specific function to perform. Mitochondria are responsible for the production of cellular energy, ATP, in virtually in all plants and animal cells, and because of which they are often referred to as the powerhouses of the cell. The chloroplasts are organelles found in the green tissues of plants which are involved in the absorption of light energy, synthesis of carbohydrates, and evolution of molecular oxygen. The sum of these three processes is called photosynthesis. Through photosynthesis in chloroplasts, organic compounds are formed from inorganic raw materials. The proteins in the cell are synthesized in ribosomes, and all the important proteins required for metabolic activities and other functions of the cells are synthesized either in free ribosomes or ribosome attached to the endoplasmic reticulum. The nucleus, on the other hand, is considered as the most essential and integral part of eukaryotic cells, as it stores all important genetic information in its DNA molecules, which can be passed from one generation to another. It controls many important cellular activities and is perhaps the single dominant structural features of most of the eukaryotic cells.